Hi, everyone, and welcome to episode 730 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I'll be joined by our guest, Dr. Laura Kiessling, to talk about life and science. This episode is made possible with support from our sponsor, New England Biolabs. Listeners, are you curious how molecular biology is moving forward around the world? Hear from scientists who are driving discovery and expanding applications of existing technologies on the New England Biolabs podcast, Lessons from Lab and Life. Host Lydia Morrison interviews revered scientists working in genetics, synthetic biology, diagnostics, molecular cloning, and other fields. Get ready to explore the cutting edge of science and learn what these remarkable scientists have to offer. Don't miss out on the opportunity to hear exclusive stories about the world of laboratory research. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And today, our guest Laura will share stories from her own life and research. So listeners, get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Laura Kiessling. So, Laura, welcome to our show today. How are you?
Good, and thanks for having me.
Well, we are so excited to have you joining us today and to learn more about you and your work. But before we get into the details, let me first start by telling our listeners a little bit more about you and how you got to where you are today. So, listeners, Laura is the Novartis Professor of Chemistry at Massachusetts Institute of Technology. She received her bachelor's degree in chemistry from MIT and her Ph.D. in chemistry from Yale University. Next, Laura was awarded an American Cancer Society Postdoctoral Fellowship to conduct research at California Institute of Technology. She then served on the faculty at the University of Wisconsin-Madison until 2017 when she returned to MIT. Now, Laura has received numerous awards over the course of her career. She was the first woman to win the Tetrahedron Prize for creativity in organic chemistry or bioorganic and medicinal chemistry, and she's also received the Breslow Award of the American Chemical Society, the Centenary Prize from the Royal Society of Chemistry, the Gibbs Medal from the Chicago chapter of the American Chemical Society, the Vilas Distinguished Faculty Award from UW-Madison, and others. In addition, Laura is an elected member of the National Academy of Medicine, the National Academy of Sciences, American Academy of Microbiology, American Academy of Arts and Sciences, and the American Philosophical Society, as well as an elected fellow of the American Association for the Advancement of Science. She's been awarded a Guggenheim Fellowship, an American Cancer Society Fellowship, a MacArthur Foundation Fellowship, and also an Alfred P. Sloan Foundation Fellowship. And she is the founding editor-in-chief of the journal ACS Chemical Biology. And today, Laura, we're excited to get to know you both as a scientist, but also as a person. So can you tell us first what you like to do when you're not doing science?
Well, there's a lot of things that I like to do. So I will say one thing I do pretty much every day is work out. And that could be a lot of different things. It could be rowing or kayaking or biking or lifting weights. I also like yoga, and I like cooking. I like art. I like camping and hiking. So lots of different things.
That sounds phenomenal. It sounds like you've got a lot of interests and hobbies, and you mentioned art specifically. What kind of art?
I really like modern art and maybe art that's not so representational, but I like all kinds of that, like from paintings and prints and sculpture. I have some really cool science-related art in my office. I have an abstract periodic table. I have a kind of paper sculpture that looks like, I would say, a microbiome community.
Oh, cool.
I like a lot of different art.
I think science can be very aesthetically appealing. There are lots of different things that you can represent in art. So I think it's so cool that you combine those passions there.
Yeah, thanks. Sometime you can visit my office and see the eclectic collection of things that I have.
It sounds amazing. And I know you're doing amazing science as well. So, Laura, for someone who's not very familiar with your work or maybe outside of science altogether, how do you describe what you do?
I love carbohydrates. I especially love the carbohydrates that are on the surface of cells. So what people think about when they think about cells is mostly about how they're encoded by nucleic acid, DNA and RNA. And on the surface of them, what I think is not as well understood is that there is this kind of forest of carbohydrates. It's a conserved feature of every cell on the planet. And we don't know as much about what those carbohydrates do as we do about, let's say, proteins or nucleic acids. And that's what fascinates me.
Very cool. Yeah, I think when people hear that just the term carbohydrate, they think of it more from a nutrition perspective, not all these different functions that they have within our bodies and within cells. So I'm excited to dive into the details here, but I know science can be difficult some days. So we love sharing motivation and inspiration with our listeners out there who might be having tough times themselves. So Laura, do you have a favorite motivational quote or maybe something that really motivates you in life in general?
I love this one quote. I think my mom told me about it, but actually I think is also the motto maybe for NASA, ad astra per aspera, which means to the stars through hardship.
Oh, I like that. Why does that resonate with you particularly?
I think because science is sometimes hard. It's hard for people to make rapid progress. But when you're aiming for finding out new things, that is the stars, it's really worth it. But the other thing that's really motivational for me is when I just think about how lucky I am to be a scientist, because we get to think about how the world works and we get to try to pursue those ideas. And regular people that work regular jobs pay taxes, and some of those tax dollars go to us if we're successful. So to me, I am one of the luckiest people on earth to have that privilege. So I don't want to emphasize the hardship too much, right? Because it's really an incredible privilege to be a scientist.
Absolutely. And I think it's so exciting to be on this path of discovering new and exciting things about the world. And I know it can help to have these not only motivational sayings and forces to keep you going, but also motivational figures or people that you look up to. So for you, Laura, were there particular people, maybe mentors, role models, people that helped you get to where you are today that really rise to the top of your mind?
Yeah, I think at different times, I'm sure everyone had teachers that have been motivational. I think back to my maybe 6th and 7th grade math teacher who allowed me to learn math on my own. But then certainly when I got to undergraduate, I had, again, professors that were really motivational. I started as an undergrad at Wisconsin and then went to MIT. And so at MIT, I was really excited by George Whitesides and Barry Sharpless. Those were my organic professors really early on. And then, of course, my advisors that I had who were my mentors, Stuart Schreiber, Peter Jervin, and Bob Grubbs. Bob wasn't really an advisor. He just was a huge mentor to me. And now it's peers that I look to and get inspired by. I could mention Jennifer Doudna, a Nobel laureate from a couple of years ago, and Carolyn Bertozzi, last year's Nobel laureate, but also friends like Sangeeta Bhatia and Deb Hung. So there are so many people for me. There's not like one or two people I can just point to.
Definitely. I think having this sort of constellation of role models and mentors and colleagues who really inspire you can be wonderful as a scientist. And you mentioned already that in 6th and 7th grade, you were sort of starting to get interested in math, maybe starting to look towards this particular area of study or of a career. So what really shaped your path as a scientist and where did it begin?
That might sound true, but I loved math and I was trying to figure out what I could do with math. So even though I took chemistry in high school and I loved my chemistry teachers, I really did not like chemistry. I really didn't like it at all.
Me neither.
Yeah. So I didn't think I really liked it. But when I started undergraduate, someone said to me, well, what if you want to be a doctor? And I thought, I don't really want to be a doctor, but I don't know that I don't want to be a doctor. And so I took chemistry. And I started to really love it, but I still thought I was going to major in something else. Like when I transferred to MIT, and I can explain how that happened. But when I got here, I was assigned a chemistry advisor, even though I thought I wanted to do physics or something else. And I took organic chemistry, and that's the chemistry of life. And it just completely turned me into a chemist.
Oh, I love that. You left a little cliffhanger there about that transfer to MIT. What happened?
Okay, so this shows that I am kind of a nerd. When I was at Wisconsin for spring break, a bunch of us decided that we were going to go to MIT. We were going to go to Boston for spring break. One of my friend's sisters went to MIT. And so it ended up just being two of us that came out, not even my friend whose sister was here. But I love meeting all of these women that were doing science. So even though University of Wisconsin, fantastic place, I loved it there. But in my classes, there were very few women. And when I came to MIT, even though there was maybe only 20% of the class was women, they were all women who love science. So I just decided I wanted to transfer. So I just went into the admissions office and said, can I have an interview? And they said, have you applied? And I said, no. And they said, what are your SAT scores? And I said, oh, I never took them. I really didn't need to at Wisconsin. So anyway, I got an interview because I had a friend who worked in the office. I just met her. So she got me an interview, and then I applied and I got in and I came. So one of my friends said, don't visit any other schools while you're on vacation.
Who knows where you'll end up?
Exactly.
I think that is amazing, though. I think it really shows initiative and sort of a willingness to make a change, which I think is really important in science when you do sometimes have to make these dramatic pivots. So then, Laura, what happened next in your career story?
So when I was at MIT, I mentioned that I became really captivated by organic chemistry. And then I started working in the lab of Bill Rausch, who was an organic chemist who builds complicated molecules. And I say complicated, but they're really molecules that nature gives us. So I was really interested in learning how to build molecules. And then I pursued graduate studies to do that, to learn more about how to build molecules effectively. And I'll just stop for a second because you might wonder, why build new molecules? And I should say that organic chemists are the ones who make all of the small molecule drugs that we take. They also are really critical for building things like antibody drug conjugates. They're the ones that make compounds that have never existed before on Earth and you can use for valuable things. They're also really, organic chemists are the people that help make new kinds of polymers and new materials. So that's a really powerful thing to be able to make something new. So that's what I went to graduate school for.
I think that's so cool. You're really selling it here. Now I want to do it.
Oh, good. It's so exciting.
Absolutely. And you decided to then go to graduate school, like you had mentioned. You got your Ph.D. from Yale University. What was that experience like?
It was a really exciting time to be in the Yale chemistry department because they had recruited some amazing people. And then my advisor was at the early stages of his career, Stuart Shriver. And it was really exciting to be able to be in this really young group and get involved in the projects. I worked on making a natural product, again, something that's found in nature that cuts DNA via a very, very cool mechanism. So that was a really exciting time. And because the molecule I was making cut DNA, it got me really interested in understanding more about the biology of nucleic acid recognition, how you recognize DNA, how molecules act on DNA. And so I went and did postdoctoral studies with Peter Dervin at Caltech.
Very cool. And then you had this serendipitous reunion going back to Wisconsin-Madison. So what really got you there for your faculty position?
I went and interviewed at a lot of places, and I was not going to go back to Wisconsin. I thought it was so boring. I grew up in a really small town of less than 3,000 people, And when I was growing up, it gave me, in retrospect, a lot of great opportunities, but I really was a little bored in school. So I just thought it would be not very exciting to go back to Wisconsin. But of course, I really hadn't been at the university very long. It was such a welcoming place. It's a place where you can do really amazing science and be supported by your colleagues. And it's just an incredible place to do science.
Absolutely. And you remained there for a number of years before making the jump back to MIT in 2017. What was that like for you, having this another big transition in your career?
So where I am now, MIT and Kendall Square is just such a mecca of science. You go to a coffee shop and everyone is talking about, I don't know, principal component analysis or spectroscopy or whatever it is they're interested in. It's really great. And one of the things about switching places is it just offers opportunities to work and engage with new kinds of scientists. And one of the kind of cool things about being here is I have all these incredible collaborators that are fundamentally new. So that it allows you to just move your science in areas that are just really different. That's been incredibly exciting.
Absolutely. And you've got some really cool, novel, cutting edge projects going on in the lab right now as we speak. So, Laura, do you have one in particular that's maybe your favorite at the moment or that you're most excited about that you want to tell us about in a little bit more detail?
I think that you can tell from all of my answers so far, I have a hard time focusing on one thing. I shouldn't say focus is maybe the wrong word, but as a scientist, what I like to do is bring together really different things. So what I'm excited about is always the latest thing. I can say a couple of things that are going on in the lab that I'm really excited about. One of the things we've been doing is the tools to look at and image sugars are not as well developed as they are in other areas. And one thing we like to do as scientists is see things, see how things are made, see where they go. So being able to see how the sugars that are on the surface of your cell are built and where they go at different time points is something that we're really working on. And we've been working on that in the case of bacteria, especially so that we could diagnose and detect bacteria that cause disease. We've been especially focused on Mycobacterium tuberculosis, which causes tuberculosis. And people don't think about this so much in the US, but TB kills over 1 million people every year. Before SARS-CoV-2, it was the number one infectious disease killer.
Oh, wow.
So new ways to diagnose and treat it are really needed. And so we've developed some new ways to image the sugars on the surface of Mycobacterium tuberculosis. And I'm going to go to South Africa in October and bring some people from my lab, and we're testing these out on patients so that especially we would like to be able to detect TB in little kids because it's hard for them to cough up the sputum that you normally need to detect it. So that's one really practical thing that I'm excited about. But the other thing I'm really excited about is, I would say, a kind of holy grail for the field, at least something that I've been thinking about for a long time. So the reason we know so much less about carbohydrates is because they're not encoded, obviously, in nucleic acids. So we can't just read a sequence of nucleic acids and know what carbohydrates a cell is going to make. But if we could do that, it would be amazing because the tools that we have to modify proteins and understand the genome are so powerful. So we've been thinking about how to relate what carbohydrates are there to the nucleic acid sequence. And so we're just starting to develop some tools to do that. And I'm really excited about that as well.
That is phenomenal. So thinking about the big picture, if you were to solve this holy grail, what kind of doors would that open for you?
So the carbohydrates that we put on our cells, I should take a step back because I really didn't explain why they're so important. So first of all, every single one of us began with a protein-carbohydrate interaction. Fertilization is mediated by a carbohydrate on the surface of the egg that's recognized by a protein on the surface of the sperm. You know, when we figured out what carbohydrate that is, and this isn't my work, it's the work of Andela and collaborators, 2013.
Wow.
Right? Amazing. And we know that carbohydrates vary between cancer cells and normal cells. Carbohydrates, their localization and identity changes as cells develop and undergo differentiation. I know you studied neurobiology, and they're actually really, really important in the brain, but we have so much to learn in this field. So this kind of relationship between nucleic acid and carbohydrate identity, if we could develop that further, and the computational approaches are so powerful now, I think combining experimental and computational approaches draws us a lot closer to that. That would be transformative for many different fields.
Definitely. And Laura, these are super exciting projects. So thank you for sharing them with us today. And listeners, if you are looking to hear about even more exciting science, New England Biolab's podcast, Lessons from Lab and Life, is your source for staying informed about the most up-to-date molecular biology breakthroughs. Host Lydia Morrison interviews prominent scientists and delves into their research to uncover how their findings affect society in meaningful ways. Tune in to learn from the experts and get a glimpse of the cutting-edge advancements being made by scientists around the globe. Don't miss out on this opportunity to gain new knowledge and stay informed on the latest discoveries. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And I know we talked about some of the fantastic work, Laura, that you're doing in the lab, but I know science isn't always happy days. There are struggles and challenges and failures that you have to work through. So do you have an example of one of your own challenges or failures that you could tell us more about and kind of walk us through how you worked through it?
Yeah. When I was a graduate student, I told you I worked on building this molecule. And it was really exciting. It was really competitive. A lot of labs were working on trying to develop a route to this molecule. We had a route that was really cool, and it seemed to work really beautifully. And we were going along doing additional studies. And I went back and looked at some of my data because some of the things we were doing just didn't make sense. And it turned out that I had made a different isomer of the product than was found in nature. So I had made something that wasn't what I was trying to make. But we had already published a paper describing that we'd made this structure. It just turned out that it wasn't distinguishable at the stage that we published the paper. But if you kept transforming the compound, then you could tell the difference between the different isomers. So just the tools we had to figure out what isomer you had weren't good enough early on. And it never occurred to us that it would go the wrong way because of all the data in the literature. So I figured it out and I had to go in and tell my advisor. And I felt like a complete failure. I felt like I would never be a scientist because I just wasn't careful enough. I wasn't good enough, all of those things that you would think. And I went in and talked to my advisor, and he was so kind and so supportive. I wouldn't be here if it weren't for Stuart Schreiber, just not losing faith in me as a person. I don't think he knew how much that meant to me at that time, but it was hard because other people in the lab were probably not, but I felt like people were almost gleeful because there's a lot of competition in some of these labs.
Yeah, I completely understand that sort of feeling of one-upping each other in the lab is...
Exactly.
So how did you get through this tough time? It sounds like you had that conversation with the advisor. He didn't yell, which is always a good start.
He told me to go to talk to another professor in the department who also was working on the compound. So I did. I was very, very depressed. It was like the worst time for me. But I guess I made it through it. I don't know exactly how, but it made me a better scientist in terms of being skeptical about things, trying to be very careful. And at the same time, it also taught me that being wrong, it's not the end of the world. People can make honest mistakes in science. And sometimes I think about, there's a lot of controversy now about scientists who are being unethical or pushing results. But at the same time, sometimes people just make mistakes because science is hard to do. And so I think that we shouldn't sanction people too much who make honest mistakes. Going in and correcting the literature when you do make a mistake is really, really important. So it taught me a lot of those lessons. And it just also taught me how important it is as a mentor to make sure that people know that one thing that happens in your career doesn't influence the rest of your career. We're in it for the long game. So you can't take one thing that happens to you as being indicative of who you are or what you can do.
Laura, I think that is such an important message to share with listeners. And I'm so glad you shared this story. And I think just keeping going is a really important trait or maybe strategy in science. Don't let these failures, these difficult times be career enders, right? You can recover from them. You can fail early and then learn those lessons and move on. I think that is definitely an important thing for people to learn out there when they're just starting on their career paths themselves.
I mean, I think I was too hard on myself. We shouldn't be hard on ourselves in that way. Obviously, learn from it, but it's okay to fail. It's okay. You can go on and do the next thing. Again, it's the long game that's important. There's no one career defining thing.
Absolutely. I think that is so important. And Laura, we don't just want to focus on the struggles and the challenges. I'd love to share some of your successes with our listeners as well. And I know you have quite a few to choose from. So do you have a favorite success story you'd like to share with us?
One of the things that's great about being a PI, running a lab, I should say, PI stands for Principal Investigator. But I think that we get to have wins on two sides. One is science, and one of the most exciting things is finding out something new about the world that no one knew. One of my favorite things that we did, and we did this a while ago. Actually, there's a bunch of things now that I think of it, but I'll say one of my favorites is we were studying this enzyme that works on carbohydrates. And in studying it, we found a completely new role for vitamin B2. And that was kind of cool because it had been studied for so long and everyone thought they knew how it worked, but we found a new way that it can work to make a reaction go. So that was neat. And then something else that I've really been excited about is, again, we did this a while ago, but I think it's been really interesting that in chemistry, one of the things that people have been doing are trying to, instead of just inhibit a protein, remove it by causing it to be proteolyzed. And these kinds of molecules are called PROTACs. They've really been pioneered by a number of different labs, Ray Deshays and Craig Cruz. And these molecules are cool because, again, you're destroying the molecule. You're taking it out of the game, basically, so that the protein no longer has its activity. And that can be really important, like if that protein is driving tumors, for example. So what we had done is actually in the 90s, we had one of the first examples of using proteolysis, not in the same way these other groups do, but we had recruited a cell surface protease to cut this protein that's involved in the inflammatory immune response off of a cell. So I think that's kind of cool just because it was a new strategy for regulating the activity of a protein.
That's really interesting. So then how does your lab celebrate some of these big successes?
Some of the cool things that we're doing now, we go over to the Muddy. The Muddy Charles is this little pub on the campus of MIT. So we can celebrate that way.
That sounds amazing. I think even just taking a moment to recognize these successes, acknowledge the hard work and the amazing discoveries that you're making, I think that can even go a long way, even if there's no sort of ceremony surrounding it.
We have other traditions, but what has been going on right now is volleyball. MIT has an incredible volleyball summer tournament going on.
Oh, really? Are you much of a volleyball player?
I won't say that I'm good, but I will say that I have played with my team here.
You've got the bruises to prove it.
Yeah, actually, I do have bruises.
Absolutely. I think getting out of the lab, taking a break from thinking about the science can sometimes be really refreshing and really helpful when you're working on a difficult problem. And one of the ways we encourage people to do this is to read and read broadly. So do you have a book recommendation, Laura, something that you've read and really enjoyed that you can tell us about and recommend for us to add to our reading list?
This is going to sound really trite, but I loved Lessons in Chemistry. I don't know if you've heard of this book.
No, I haven't.
It's a fiction book. It's fantastic. It's a very fun read, but it has a seriousness in parts to it too. It's about a woman. She kind of is in her 20s to 30s in the 60s. So a while ago. And all she wants to do is to be a chemist. But she's really thwarted by society from doing that. So she, through a series of odd events, she has a cooking show where she describes the chemistry of cooking and then kind of motivates all these other women to take themselves more seriously as women. And she has them all taking notes, and she describes in detail about the Maillard reaction in meat browning. It's just fantastic. And she's also a rower, which I was, so I could really identify with her. But even if you can't, it's a really fun read just about how we need to create spaces for people who love science, who aren't traditional, and how we can do that and how those people can get through difficulties to ultimately succeed.
Oh, I love that. This sounds like an excellent book. We will add it to our reading list on our website for our listeners out there, Lessons in Chemistry. And Laura, thank you for sharing that. And we've talked about some different aspects of your career in your life. And one of the things we haven't really touched on yet is travel. And I think there are so many amazing opportunities to travel as a scientist all over the world, whether it's for meetings and conferences, to meet with collaborators, etc. So do you have a favorite place that your science has taken you?
So I have been a lot of amazing places. One of the funniest things was my daughter in 5th grade. They were making a zine for their class and they had to write a rant. And her rant, which got posted in the board at her school, was, "I hate chemistry. It's so boring. The only thing good about it is the travel."
Oh, wow.
That was tremendous. But I think one of the most memorable trips was I was in Japan with my husband, who's also a scientist, chemist, and our daughter at that time was 14 months old. And we were on a tour bus with the whole conference where we had been visiting the city and we had to get diapers. And so the whole conference bus stopped at a grocery store so that we could buy diapers. And my friends, other scientists on the bus, could not stop teasing me about how I had stopped a whole conference bus to buy diapers.
Pit stop. So how was the trip?
It was amazing. We took some time off before it to travel around. And people, I guess, don't normally take very small children traveling around Japan because we had many interesting experiences. Like when we showed up at a restaurant, and they were just like, no. They kept saying, for two people. And we're like, no, for 3 people. And then finally, we realized, I guess you don't want us here.
There is clearly a third. Yeah.
I think they sent us to the wrong restaurant. It was too fancy for people with a small child.
Oh, well, that sounds like an amazing experience. And I think being able to go to all these different places is definitely a perk of the career in science. If our listeners are able to make it over to Japan, was there something you saw or did on your trip that you think they have to add to their itinerary?
The Moss Garden, which is outside of Kyoto, is the most incredible place. You have to make a reservation to go there. And you go there and you have to do some Buddhist chants. And then you write in calligraphy, which took me a very long time because I wasn't very good at the calligraphy. You write the characters. It's very meditative. And then the moss garden, I can't describe it, but it has so many different kinds of moss. It's so beautiful and different than any other kind of garden that you've been to. So that was an incredible experience. And one thing I'll say is, even though people weren't used to having small children traveling, the Japanese people were so kind and accommodating. It was really wonderful. And random people that we'd met then sent gifts to Kira, our daughter. So it was great.
Well, this sounds absolutely amazing. And I think you're right. These opportunities to travel really open up your eyes to how things are in other places and connect you with scientists who you might not otherwise meet because they are from around the world. And I think the people in science are part of what makes it great. There are so many amazing, creative, funny people that I've met who are scientists. And this goes against some of the stereotypes that I think people still have about scientists and life in science. So we love to try to break those on our show by talking about some of these human moments, the traditions, the fun, the funny things that you do when you're together, because I think these help people realize that scientists are just regular people with really cool jobs. So we'd love to share some of your stories if we can today. You mentioned this tradition of volleyball, which I think is amazing at MIT. Are there other traditions that you have currently, or maybe that you experienced in a previous lab, or just a funny story you'd like to tell us about today?
This happened when I was a postdoc, and I worked with Peter Dervin, who's a wonderful scientist, but pretty serious. We always thought in the lab that Peter didn't notice things that we were doing. So one of my friends in the Dervin lab was joking around, and she said that she had a crush on this professor, Paul Bartlett. And she was just going on and on about how he has great eyes and he's so cute. And so we thought this was so funny because his picture was, he'd won some award. And so we took his picture and we wrote next to it, this person's name was Jumi. Jumi, I can't stop thinking of you. Love, Paul. And we took and made copies of this and we put it everywhere in the lab. So she would open her drawers. There would be that picture. She would pull down her hood sash. There would be that picture. It was just everywhere in the lab. So then we went to group meeting and Peter had just gotten back from Berkeley, which is where Paul Bartlett was. And he said, oh, I just got back from Berkeley. It was great. I saw this person. They're doing amazing work. And I saw that person. They're doing amazing work. And then he turns to Jumi. He goes, oh, and by the way, Jumi, Paul says hi. And we just lost it because it was just so anti everything we thought about our lab. And that's the thing about labs. The advisors, you don't think they're paying attention? They're paying attention.
Well, I love that your lab had a lab idol sort of in the Paul Bartlett.
That's why I thought I would tell that story. We're just people. We goof around and joke around and have the same crushes, think somebody's cool, somebody's not cool, whatever.
Well, I think that sense of humor goes a long way in making the lab just a fun environment to work in, but also contributes to the creativity and the willingness to go out there and do some of these big, hard things in science. And I think that's so important for solving the big challenges we're facing in the world today. So, Laura, if we took away the things that normally hold you back, whether it's funding, staff, technology, time, feasibility, whatever it might be, and took all of those barriers away and you could do anything and had all of the resources you could dream of, what is the one question you would want to answer most?
I am really interested in this question of how to relate genomic information to what carbohydrates appear on the cell surface. And I know that sounds so abstract, but like I said, I think it would be transformative for fields ranging from neurobiology and cancer to microbiology and the microbiome. It's something that I would really devote a lot of time to because it could then spin off whatever interest really you had. Like, again, if you were really interested in understanding cancer better, or if you really wanted to understand how the brain works, those are all big questions that just could be enabled by the tools to do what I was talking about.
Definitely. So what do you think is the biggest barrier right now in developing these tools?
We need more data on both sides. We have a lot of sequencing data, but we haven't related it to what carbohydrates are there. So we have to look at what carbohydrates are present on the surface of the cells at different time points and then relate that. And one of the things we've been really working on is to think about, there are a bunch of human proteins that recognize carbohydrates. We don't even really know what cells those proteins recognize. You know how people want to explore the stars or want to explore what's under the sea? We haven't even explored this aspect of what's on our cells. That is, I think, an all-encompassing question just because it has so many ramifications for so many different parts of biology. And I should mention also that in disease, the viruses use a lot of these carbohydrate binding proteins to invade us. So protein carbohydrate interactions are critical for SARS-CoV-2 invading our cells, for influenza virus invading us. There are so many diseases that also could be basically mitigated by understanding this better.
Absolutely. I think you're so right there. There are still so many unanswered questions. This cell surface is still an exciting frontier in science with a lot of work to be done. So you've got your work cut out for you, Laura. But I know for listeners out there who might be starting their scientific journey, it can be tough. We shared some of the stories and some of your experiences today, but I'd love to end with advice if we can. So is there one piece of advice, Laura, that somebody gave you at some point in your career that really helped that you can pay forward to our listeners today?
This, again, I think is something that maybe other people have said on this show, but I'll just say it, not being afraid to fail and not taking a temporary failure as a permanent setback. To me, those are just the most important things in making your way through science.
Absolutely. And I love this point that you made about it sort of being a transient or temporary setback, something that you can work through and completely move on from. These failures aren't going to sort of follow you for the rest of your life in most cases.
One thing doesn't define you unless it's an ethical breach. That, I think, could define you.
Well, Laura, thank you so much for sharing this advice. Is there any other last message or a last note of inspiration you'd like to leave our listeners with at the end of our conversation today?
One thing that I don't know if we talk about enough as professors, we talk a lot about discovery, and that is certainly why we get into it. But one thing that I don't know if we share is how cool it is to see students go from not knowing something to really making strides in learning it and applying it. It's one of the most rewarding things about our job. And so watching like someone come in as a beginning undergraduate or a beginning graduate student, and then watching them as they leave is just one of the privileges that we get to have as faculty. And at least if we have a little part in helping people make that journey, it's very rewarding.
Absolutely. I think this mentorship and training the next generation of scientists is a really valuable and sort of invigorating part of the career. So I'm glad you brought that point up as well. And if our listeners want to learn more about you, your lab, and the amazing work that you're doing, what is the best way for them to do so, Laura?
My website is a good place, which is kiesslinglab.com. And the other place to learn about what we're doing is on Twitter, formerly known as Twitter. And I'm at Chemical Biology.
Fantastic. Well, listeners, definitely get connected with Laura. Check out her website to learn more about their projects. And Laura, it's been such a pleasure to have you on the show today. Thank you so much for your time.
Thanks. It's been fun to talk with you.
Well, it's been great to have you here, listeners. Wonderful, as always, to have you here with us as well. And we hope you join us next time for another episode of People Behind the Science.